Yeah, hi everyone, thanks for that. Sorry I won't be talking about fishery longevity like Mark said, but today we're going to talk about some work we've been doing looking at the vertical distribution of cisco and Lake Superior during their spawning aggregations and some potential implications for evaluating our management benchmarks. So, thanks. Before I go on, I'd like to thank all my co-authors. I'd also like to thank the Great Lakes Restoration Initiative for providing funding for my position, as well as the RV Hack Noise crew through the years for doing all these surveys. I'd also like to thank Wisconsin's Lake Superior Commercial Fishers, as well as our partners at Red Cliff, Bad River, USGS, and the U.S. Fish and Wildlife Service. So, quick introduction, Wisconsin's seen a recent increase in commercial harvest of cisco. So if you look at the figure on the right, you can see essentially a regime shift starting in about 2009, where harvest increased. And that's been the result of the emergence of a roe market. So these commercial fishers are targeting this Lake Superior gold to bring to market from these fecund females. So in response to that increase in harvest pressure, Wisconsin did implement a harvest quota in 2016 with the goal of protecting this stock from overexploitation. So one management benchmark that's been proposed for a sustainable fishery comes from Stockwell and co-authors to limit exploitation on large females to roughly 10 to 15%. And to be able to evaluate that management benchmark, it does require knowledge of the stock size. So starting in 2016, Wisconsin waters have also started to rely on hydroacoustic surveys as well. Generally, these are paired with midwater trawls or suspended gill nets to get an idea of the relative species abundance as well as the relative sex abundance. So since our research vessel, the RV Hack Noise, is an old commercial gill net tug, we rely on a suspended gill net survey because that's what we're effective at lifting. So our gill net survey that we've been conducting consists of paired top floated and bottom set gill nets. However, because cisco are believed to be pelagic spawners, we do only use the top nets when apportioning our echograms. However, it's been noted in the past by Yule and co-authors that there does appear to be dynamic distribution during these spawning aggregations where males become more bottom oriented as the season progresses. And given this survey design layout, we thought it provided some interesting ways we could look at the data. So we wanted to look at a few things. One, we wanted to determine if we could identify any sex selective bias within males and females, if they're more likely to be caught in the top or the bottom nets. Then we wanted to explore how this potential bias in catch distribution might affect the sex ratios that we're using for evaluating our exploitation. And last, we also wanted to look at the effect that maturity might have on this vertical distribution as well. So moving into our sampling design, these gill net surveys, again, have been conducted since 2016. And I'd like to make it clear that they are temporally separated from our hydroacoustic surveys. So when our boat's in hydroacoustics mode, it's not setting or lifting nets. So generally, we get out either before and or after our hydroacoustics window. So it consists of paired top and bottom net sets. The top net's floated about 10 meters below the surface. The bottom net's fished down the bottom in anywhere from 30 to 80 meters of depth. And we set those nets 100 to 400 meters of each other. And then we set two paired locations per 10-minute grid cell. It consists of graded mesh panels ranging from 1.5 inch mesh up to 4 inch mesh to give us 600-foot long gangs, and they're fished overnight. So when we lift these nets the next day, we're taking some information, net location, as well as if these fish were caught in the top or bottom, the associated mesh size, and then we're taking physical data, so total lengths, weights. We're identifying sex and maturity, and if those can't be identified externally, we do dissect fish as well to get a proper read. But sometimes there's just so many fish that we do have to take counts as well, depending on how saturated the gear is. So moving into our heuristic DAG, or our directed acyclic graph, this is a representation of the things we think makes up the system we're trying to identify and how they're all related. So causes flow with arrows, but associations do flow in both directions. So moving into the data that we have information on the total catch, which is the sum of the bottom and the top net catch. We have information on the sex, so if these are male or female fish. And then we did identify maturity as well, so if these fish are immature, green, ripe, or spent. And with that maturity information, we also defined post hoc what we call a spawning period. So if we had any spent fish in the survey, we called it a P of spawning. Otherwise, it was assigned a P of pre-spawn. And then we have information on sampling events, so dates and the 10-meter grid cells. Last, we also identified these unknowns, potential covariates that might be influencing the progression of the spawning period, so things like water column temperature or cumulative cold exposure. We don't have any information on this, but we think this might be playing a role in this system as well. So moving into our first objectives, to look at the sex distribution, we relied on aggregated binomial regressions. And for that, we're solving for this probability of being caught in the top net. So as that value of p approaches 1, that means that a fish of a given sex is more likely to be caught in the top net. Conversely, as it approaches 0, it means they're more likely to be caught in the bottom net. We defined this null region covering roughly the 2 to 1 to 1 to 2 odds range. And we really wanted to quantify anything that was above that 2 to 1 odds in either direction. We called that bias. So taking the models from that first objective, we then used them to simulate using a known sex ratio, because out in the field we don't actually know what the true sex ratio is, but we can get an idea of how this net distribution might influence the sex ratios through simulation. So we pushed 1,000 males and 1,000 females through each of our models. And we again calculated what the expectation of the top net catch would be. We defined a null region ranging from 1 to 2 to 2 to 1, females to males, and we were concerned with identifying how much the sex ratio might vary from that true known value if we're only considering the top nets. And last, looking at the effective maturity. For this, we weren't as concerned with identifying bias, so we were just looking for the support in those aggregated binomial regressions, either above or below that p of 0.5. So moving into some of the results that we found. Shown here first is the workup of our catch data. So across the top row is our catch in the top net. Across the bottom rows are the catch in the bottom net. And moving from left to right across this figure are the years. And one thing that might stick out to you in some of these sampling events is those blue bars on the bottom are pretty thick. So that's what Yule and co-authors identified. There appears to be males aggregating on the bottom, but if you look at those red bars, for the most part, they look pretty even from the top to the bottom. So when we consider maturity as well, so shown here is the proportion of that catch for the top and bottom nets by the assigned maturity. And what might be sticking out to you here is that bright green, that's maturity of spent. So these were spent females, and they look to all be in the top nets, but if we look at the bottom nets, we're seeing a lot more orange in place of that green, and those are ripe females. So just by looking at the data, it looks like the spent are distributing on top, the ripe are on the bottom. When we look at the maturity for males, we see a whole lot of orange, so ripe males all over the water column. So if you might recall earlier, I said we did take counts depending on the degree of gear saturation. So shown in gray here are the counted fish. In some of these sampling events, there's a much bigger picture of just the amount of fish that were caught in those bottom nets. But it's not always the bottom nets. If you look in the middle plot at the top, in 2019, you can see there's still some gray bars up there as well. We used a method called mice to impute what these potential sex and maturity combinations would be. So this is just one instance of a realization of that. And this is what we use to move forward to evaluate our objectives. So shown here is that distribution of the probability of being caught in the top net. So again, top half of the graph represents more likely to be caught in the top. Lower represents more likely to be caught in the bottom. Shown a dashed line where that p of 0.5 is, or the 1 to 1 odds. And then those point distributions, the dot represents the median, as well as the 50% and 89% quartile. And females are in red, males are in blue, and left to right is that p of pre-spawn to p of spawning. So you can think of the time period progressing from left to right. And hopefully what sticks out is that in addition to that blue distribution going lower as time progresses, we're also seeing that red distribution go lower. So females are also more likely to utilize the bottom as the spawning period progresses as well. And if we evaluate at 80% posterior support for these hypotheses, we do get significant results. So earlier in the year, females much more likely to be in the top net. Later in the year, males much more likely to be in the bottom net. So how can that cascade through to the sex ratios that we're using? So shown here is the distribution from our simulation. That dashed line represents the true 1-to-1 that we put into these models, and those dotted lines represent the 2-to-1 to 1-to-2 female to male. And at the top is that distribution for that P of pre-spawn, and you can see that median of the distributions right around the dotted line. So about 2-to-1 females to males if we're relying on the top nets earlier in the year. But as the season progresses, we see that distribution shift to about 8-to-1 females to males. So same number of fish, but if we're only looking at a snapshot from the top, we risk assuming there are more females out there. And again, if we evaluate at 80% posterior support, it does come out with the star. Look at that. Last shown here is the distribution, assuming maturity as well. So again, on the left is pre-spawn, on the right is spawning. And going down the rows, is, you can think of it as the progression of time from the top to the bottom. So that top row is maturity of green, that middle row is maturity of ripe, and that bottom row is maturity of spent. And what I'd like to have you look at first is on that left column, those distributions for sex and maturity combinations overlap pretty well. So we see that green individuals much more likely to be in the top, ripe individuals about 50-50. But then when we move to the right column, we see those green individuals for females, it's roughly 50-50. All of the males are on the bottom across the board, but what's interesting is the ripe females much more likely to be caught on the bottom as well. So there's this paired of ripe males and females happening later in the spawning season. And then when we consider spent individuals, we're getting pretty much all the females on top for that. And again, when we evaluate at significance, 80% posterior support, we do get stars to look at. And again, the interesting one here is just the paired ripe males and females on the bottom. So a lot going on there. What does that mean to us as managers? Well, we knew there was a dynamic sex distribution. And what this means for us is that it will have implications. So given our current survey design, it will influence our estimates and likely result in overestimates of female biomass and underestimates of female exploitation if we're only using that snapshot 10 meters below the surface. So what might be causing this? We might be keying more in on just this initial staging aggregation versus the actual active spawning. So if we assume there's an associated behavioral shift, as these maturities progress, these females might be moving to the bottom to spawn on the bottom, or some portion of the spawning behavior involves the bottom. These males are following them down and potentially staying down there. Maybe they're moving back up into the aggregation. We can't tell that with this data. But when the spent females are done, it looks like they're the ones moving back up into the very top of the water column, and that's what our nets are catching. So before we assume that our commercial harvest might be creating this evolutionary response, I just want to put it in some historical perspective. So if we go back to Dreyer and Beal, which was the first confirmation of pelagic spawning in ciscos in Lake Superior, the lines of thought that they used were they found these spawning aggregations on the fish finder in the Apostle Islands in the pelagic region. We can still do that right now. They caught cisco and midwater trawls, as well as eggs in a zooplankton midwater trawl that they described as undoubtedly drifting toward the bottom. But there was no mention of if these eggs were fertilized. Anybody who's handled the ripe cisco knows that they do release their eggs pretty easily. So these fish could have been trying to avoid the gear. Hard to say now. And then the last line of argument from Dreyer and Beal was that a majority of the commercial fishing on the North Shore was done at the top of the water column. However, they did note that commercial fishers outside Marquette started at the top of the water column and then fished lower as the season progressed. That's what we're seeing in our commercial fishers in Wisconsin waters right now. And then they did note as well that in the flesh fishery on the South Shore for cisco, a majority of the harvest was taken on nets at the bottom. So they did leave the possibility open that these fish may be spawning on the bottom. So follow up, what can we continue to research that would be useful for us as managers to be able to try to correct this potential for error in our sex ratio assignments to these hydroacoustics surveys? We could explore this U, these unknown environmental covariates. So it could just be the water becomes isothermic and then these fish will utilize the whole thing. We can't say that right now, or it might just be a degree of cumulative cold exposure that's pressing the spawning period along. A lot of potential questions that we'd have for just things we can latch onto to try to correct our estimates. And since it's really hard to get out on Lake Superior in December, maybe we could rely on a method such as stationary monitoring, where we deploy a platform that's up-looking underneath one of these spawning aggregations, where we can essentially observe this for longer, so we could see, if they're moving down and staying down, are the males staying down, or are they moving back up into this aggregation in the water column? Things that would be useful for us as we're evaluating these echograms. But in conclusion, given our current sampling design, it will result in inaccurate estimates of female biomass and exploitation if we're only using our top nets, but still a lot more to learn in terms of the spawning behavior of cisco. So thank you.